Hi, everyone. So I'm here today to kind of dive into what Heather touched on briefly during her presentation, which is how the biological communities in Long Point and less so Rondo are responding to this large-scale Phragmites australis control. And I've been working in Long Point with Phragmites since around 2014, so I started looking at how wetland bird communities responded to invasion and that photo that Heather had on one of her last slides of that person standing on a ladder in Phragmites, that was actually a photo of me in my first year out there. So I've presented on this work for a long time. And just recently, a friend brought this column, or some of you may have heard or already read this column that recently came out in the Aurelia Matters. I imagine it made the rounds because the friend who sent this to me was from Alberta and she really doesn't study invasive species. Um, But David Hawk here, the title is kind of provocative. Is it time to rethink our battle against invasive species? And I thought this was really interesting, and I wanted to, it kind of sparked inspiration for this talk today that I'm sharing with you. So, for those of you who haven't read it, David touches on a few things, but briefly he asks us, um, or he talks about how we understand how a system works through our ecology, or at least we think we do. So we try to make plans to ensure that that system continues to work within these known values of health, that we maintain that ecological health and integrity that we know should be there. But then he asks us, what era are we necessarily trying to maintain? Are we going back to pre-industrialization, pre-colonization? Are we going, where exactly is our target? And he's kind of sums it up as therein lies the frustrating challenge to land managers. Just what is it that we're trying to conserve? And I want us to kind of just keep that in our heads while I go through this presentation today of just what is it that we're trying to conserve? And beautifully in this column, in nearly the next paragraph, as an example of the trade offs of invasive species control, he describes the project that we've been working on in Long Point and likely got this information from a paper that I recently published that was part of my PhD, where in the first 2 years after Phragmites control, we're seeing a lot of invasive European frogbit coming back into the marsh. And the words he uses here are another invasive species, European frogbit, was allowed to flourish and contaminate the area. And I think we could probably spend a lot of time even just drilling into that choice of language about contamination. But I just wanted to thank David for giving me a little bit of inspiration for my talk today. So, what I'm going to do is bring you through a kind of overview of all of the comprehensive monitoring that we've done, while also thinking about our relationship to the land and why we do invasive species management and what our goals are when we do it. So, if you've seen Erica and Heather's talk, you are intimately familiar now with Long Point and Rondo, perhaps less so, which is another sandspit peninsula on the north shore of Lake Erie. The herbicide monitoring and the vegetation monitoring that I'll be sharing with you today took place in both Rondo Provincial Park and in Long Point. And when I get into the sort of the wildlife, so the invertebrates and the birds, that's more in Long Point. And the work that I'm talking to you about today is the work that Heather mentioned that was kind of that pilot. So that began in 2016. And what I was a part of was 2016, 2017, and 2018. So just to keep that in mind, we're talking about Cranland and we're talking about the Long Point Provincial Park, not the national wildlife areas. And I want to get into a little bit of the mechanisms for why Phragmites itself is so changes these wetland habitats and affects our native species. And so I drew this schematic of what a marsh habitat looks like in Long Point. Um, and yes, I did this while productively procrastinating for my PhD. But to give you an idea, if you haven't been to the marshes of Long Point, they're coastal and they sort of exist along this water depth gradient. So what you see in the deeper standing water areas, so kind of to the left on the screen here, you have your submersed and floating aquatic vegetation, and then you have dense emergent vegetation. So you have your rushes, your cattail. And as we move to areas that have shallower standing water or saturated soils, we have sedges, grasses, and then we move into a sort of Saturated soil but less standing water, where we have a really high diversity of grasses, sedges, forbs, plants. Um, so really, the native vegetation in these marshes creates this beautiful structural heterogeneity that supports a lot of different species because there's such a wide, different amount of niches here. And in the late 1990s, we have really great information for longpoint and frag invasion. So in the late 1990s, we saw Phragmites start to expand exponentially. So when frag gets into these, begins to establish, it's capable of establishing along that entire water depth gradient. And while it may not completely displace every single native species, it will overrun them. So essentially, it looks like a monoculture. When you get in there, you might find a few native species, but for all intents and purposes, it is mostly Phragmites. And it goes from that shallow, saturated soil, dry out into the standing water. So it's taking that structural heterogeneity that's so important, all the different tall emergent veg, the open water patches, the shorter vegetation, our submersed aquatic vegetation. We're seeing all of that structural heterogeneity be replaced by a monoculture of this dense, tall Phragmites stands that you are all familiar with. And this has been documented as being incredibly detrimental for the many species at risk that and just the unique species that rely on these wetlands. So I've pulled a couple published articles here. We can see, as Heather touched on, the effects of Phragmites on blandings turtles, and it sort of restricts their home ranges. They get caught in it, they desiccate. This is some work, this middle paper here that I did, looking at how birds use invaded Phragmites habitat and found that wetland birds of conservation concern, so wetland obligate species like the rail, they were not really using Phragmites. So there may be species like red-winged blackbirds or marsh wrens in frag, but all of those species of conservation concern, they were using the remnant uninvaded patches in the wetland. And similar species like Fowler's toads, Phragmites was really decimating their breeding habitat. So we have this well documented body of knowledge that tells us Phragmites, when it's sort of expanded in these marshes, it's replaced our native vegetation. It's really detrimental to the species that rely on these marshes. And again, Longpoint and Rondo represent over 70% of the remaining intact wetlands on the north shore of Lake Erie, so on the Canadian side. These are incredibly important habitats for these species. If they are pushed out because of an invasive species, or likewise development or anything else, there's nowhere else for them to go. So when we recognize those negative effects, how do we handle that in a sensitive ecosystem? And this is where I wanted to bring it back to what are we trying to conserve? And I guess I want to talk about something maybe a little bit taboo, but inspired by Cliff, is I want you to think about what do we love? Maybe we don't say it out loud to ourselves, but I think everyone who's here who wants to protect and conserve these habitats is because on some level you love the land, you love these species, and you don't want to see them harmed. So how then does that change the way we're thinking about invasive species management? How does that affect our goals? We recognize the importance of these places. So how do we care for them? We have a relationship with these wetlands. We understand the harm. What's our relationship? What's our responsibility? And Heather touched on, or she gave a really great overview of the application to control Phragmites, so I won't be going into it. But the choice here was we see that this Phragmites is negatively affecting these species, and we understand the value and importance of this land, so we need to do something. And a large-scale invasion like the Phragmites invasion in Longpoint and in Rondo, that required a large-scale approach. So in 2016, that was the helicopters applying this glyphosate-based herbicide. 2017, 2018 was also a more ground-based application. But this was, as Heather pointed out, hundreds of hectares of Phragmites were treated with a glyphosate-based herbicide so that we could kind of knock back those invasive species populations and return the habitat that we knew was so important to species at risk and the other species that rely on these wetlands. So we have an approach and we have an objective, which is to increase or like improve the habitat for the species that use this wetland. And the work that I'm going to be sharing with you is our very comprehensive monitoring because we expect that there are large changes when we go from very dense Phragmites stands to we've treated them with herbicide, we've knocked them down. That's a fairly large disturbance in a system. So we want to know all of these connected parts, how do they respond to that? So I'm going to give you a really brief higher level overview of the story of the Phragmites management and of our monitoring, which I think is fair to say the most comprehensive monitoring program in Canada on freshwater Phragmites management. So I'll be sharing with you our studies of herbicide concentrations, how the vegetation communities responded, how macroinvertebrates, so aquatic macroinvertebrates, respond to the treatment, and how aerial insectivore birds respond, and also give you an idea of sort of the continued work that's going on in these marshes. So beginning with the herbicide treatment, this map, I recognize it's incredibly busy and I don't need you to pull out the information here, but going sort of starting from clockwise top left, A, B, C, these are just highlighting the areas that were treated each year. So A you can see is 2016, B is 2017, C is 2018. And the points are the sites where we were measuring glyphosate residue in the soil and water. We also did some additional paraphyton and monitoring, but I'm going to be focusing on the soil and water right now. And we did this, if you see in panel D, we went right in the Phragmites that had been treated, and then we systematically went out into the bay so that we could also see how far out the glyphosate residue and its associated breakdown products dispersed into the bay. And we wanted to see how long it stuck around. And as Heather alluded to, we had very promising results. So this is the concentration of glyphosate in water in milligrams per liters or parts per million. You can see in the baseline measurements for each year, so 2016, 2017, 2018, there was no glyphosate present pre-treatment. Within 24 hours, so in some instances, we were out there just a few hours after they had sprayed these sites, and we were collecting water and sediment samples. You can see the concentrations are slightly elevated within 24 hours. And to note, 2016 was the aerial application of herbicide, 2017, 2018 was more ground application. But these concentrations are still very, very low. The guidelines for the protection of aquatic life to chronic exposure of glyphosate is 0.8 milligrams per liter, so double the y-axis. And then for acute exposure, it's 27 milligrams per liter. So this data wouldn't even show up if I put those lines on the y-axis. And then another nice piece of information is that when we went out and sampled about a month later, we barely detected, there's one small detection, but there was, the glyphosate had dispersed from the, these points. And we never really captured it more than 10, 20 meters out. It wasn't dispersing very far from the point of application, and it was returning to baseline very quickly in the water. Now, similarly, for glyphosate concentrations in sediment, these concentrations were very low. Pre-treatment, the concentrations were zero. We weren't really detecting it. Within 24 hours, you see again that mild elevation. And then 20 days later, we're still detecting glyphosate in the sediment because glyphosate, this is a known property of the active ingredient, it just persists longer in sediment. So this isn't surprising, and the concentrations are still very, very low. One thing that is of interest is that in 2018, one of our transects was in the same spot as it was in 2017, which means it was sprayed in 2017 and then it was sprayed again in 2018. And we captured this in our baseline, actually. So this point here, 2018, where you can see there was one data point that had some glyphosate residue in the sediment still, that's from the previous year's treatment. So this gives us an indication that glyphosate can persist in the sediment for a year or so after treatment, although in very, very low concentrations. And in some of our different points, we sampled 2 years later, and it was typically gone back down to baseline levels at that point. So while these concentrations are very, very low and not concerning, it is something that we learned, which is if you're treating multiple times in the same area, there is the potential for residue to accumulate in the sediment, though again, very low concentrations. So we have positive results from our glyphosate concentrations in sediment and water, that the concentrations were very, very low, which is what we'd like to see. And now I want to move on to sharing some of the vegetation sampling with you. So just briefly, what we did vegetation sampling in both Long Point and in Rondo Provincial Park. And we set up 80 sites, so 40 in Long Point, 40 in Rondo. Um, those sites were evenly split between sites that were treated with glyphosate and sites that were not treated but of equivalent density. So we had sites that we were able to go back to. We sampled before treatment began in 2016, and then we've been returning to them every year after to see the differences in vegetation trajectory. So we have our treated sites, and then we're able to compare the treated site composition to the sites with dense Phragmites that were not treated. And this has given us some really interesting and invaluable results. So the other thing that I want to point out is maybe you could tell from Heather's presentation, this is an incredible amount of work. So here's photos of us out in the field in Frag. We measured the community composition. We measured the heights of these stands. We measured the light penetration. We counted all of the live stems. We measured water depth. We did a lot of measurements here. Um, and it would typically take a crew of 3 at least a week to do both if we were really like motoring through them. It is an incredible amount of work. All of this monitoring was an incredible amount of work. And we hope that it is helpful to others who don't have the resources to necessarily do this thing. So I just sort of, sometimes we just see a box plot. We think, oh, that's great. I want to do the same thing. But I do want to highlight that this does take a lot of people, a lot of hours, a lot of resources for us to go out there, for anyone to go out there and do this. And again, as Heather alluded to, so this is 2016, which was pre-treatment, and then this is 2 years after treatment, which is what I'm sharing with you primarily today. And we see in that first year, like the first growing season back after treatment, there was an over 99% reduction in Phragmites, live Phragmites stems compared to the untreated sites. And then we begin to see a little bit of a grow back following 2 years afterwards. So that's when we get to around that 95% value that Heather was sharing. So it's still incredibly effective. There's a little bit of grow back as expected. Looking at the plots that I had, there was really no rhyme or reason for where Phragmites was growing back. Some sites were shallower, some sites were deeper. I can say the one shallower site did have pretty rapid grow back from like 1 stem to like 20 stems within a growing season. So that's just some additional information here for what's growing back, but it's really incredibly effective. And this is the part that was brought up in that column and perhaps took the wind out of people's sails a little bit after those first 2 years, is that after treatment, there's fairly low amounts of vegetation those first 2 years in the sites where Phragmites have been treated and removed. And this is a photo from a site in Crown Marsh on Long Point. And you can see we had really high water level years. I remember the site in 2014 was actually meadow marsh. But here in, I believe it was 2017 or 2018, we have very high water level, very high water levels. You can see the sparse sort of cattail growing back. There's a lot of aquatic, like submersed vegetation here, but it's not a robust native like community necessarily. It's still, there's a lot of shallow open water, and it's a very stark difference from if you're driving down the causeway. The year before, it was tall, dense stands of Phragmites that blocked your view of the bay. The next year, it had been knocked down, it looked more sparse, and you could see the bay for the first time in what I imagine was a few decades. So it's a fairly, especially for people visiting Long Point, is a fairly dramatic difference. And then in the majority of our sites, they were dominated by European frogbit. So European frogbit is this small floating plant. You can see it here. It's created this mat over, so there's standing water here, there's frag stems that have been knocked down, and then there's just frogbit kind of going crazy on top. And it's this small floating plant that creates these very dense, they're just very dense mats that are challenging to walk through. They make it difficult to fish. And it was brought to Canada as a herbarium specimen, I believe in the 20s, where it got out. It's been proliferating ever since. And this is the story where we've removed the frag stems, and now there's a lot of frogbit. So it looks like we sort of removed one invasive species, and then another just came in and colonized and took over, which in some aspects, I'm sure the removal of Phragmites did improve frogbit's expansion. There was more space available, there was more light available to frogbit, but it's important to note that Phragmites and frogbit co-occur in standing water. So this is a photo of a Phragmites stand, which would be very dense, and then in the standing water, there's just a whole mat of frogbit. And I think this is something that sometimes gets lost in translation, which is these two species, they were co-occurring. So when we treat it, it reaches the Phragmites, and because Phragmites is so dense and the way that they apply this herbicide is so precise, it's actually not, it's barely reaching into the water below. So the next year when it comes back, Phragmites is no longer there, and frogbit has a chance to sort of grow in the same patterns it was growing in or expand more widely. And this is just something where I want to highlight that removing Phragmites doesn't necessarily mean we've now left space for frogbit to contaminate these sites. Um, and another important consideration is these are not two invasive species of equal damage in these wetlands. Phragmites, we know, crosses that entire water depth gradient. It creates these dense monocultures, and we have these documented negative effects for a whole multitude of species. Frogbit, on the other hand, requires a level of standing water. You're not going to find frogbit replacing meadow marsh. Additionally, the literature is pretty sparse about the effects of frogbit on native species. It likely is choking out other native submersed vegetation just because of the really dense mats. It can make fishing, as I mentioned, challenging, and it can affect macroinvertebrates, though, again, the documented effects are limited. So these two species are really not, it's not a trade-off of one very detrimental invasive species to another very detrimental invasive species. It is simply, it is a trade-off of one very detrimental invasive species to one non-native species that has less impact in our marshes. And a nice component of this is that Megan Jordan, a current master student, she has picked up where I left off. So she's been doing these surveys in 2019, 2020, and 2021. And here's her preliminary results, which is the number of invasive species at our sites is decreasing considerably, whereas the number of native species is increasing considerably. And she likes to joke that she got the good years and I got the worst years. But she's also noticed that this coincides with a decrease in frogbit, likely because frogbit needs some assistance from emergent vegetation to protect it from the wind and wave action that can jostle it. So without frag, without dense emergent cattail, it begins to sort of dissipate those over time. So we're seeing native species return. And this really highlights that we shouldn't be discouraged by the first few years after invasive species control. Invasive species management is a massive disturbance to a system, and we would expect that it takes time to recover. And in fact, we see that when we're able to do monitoring for more than 5 years. And on that note, I also had questions about macroinvertebrates and aerial insectivore birds. And this actually came from my master's work where I assessed wetland bird communities. And I noticed that aerial insectivore birds, so that is mainly swallows, birds that eat insects while they're flying, they were consistently less abundant over Phragmites stands. So for my PhD, I wanted to test the hypothesis that this was because there was less prey available in Phragmites. And this here is a map where we set up, on each one of these, we set up an emergence trap to capture emerging invertebrates. And then we also did a point count for aerial insectivore birds. And we looked at invaded, so Phragmites sites where there was no treatment, herbicide-treated sites over here in Crown Marsh, and then in remnant marsh, so cattail and meadow marsh, kind of along the water depth gradient, so that we can have a comparison of all of these. And again, I want to reiterate, so up here on the top, this is an emergence trap that was set up for the entire field season. And I just want to reiterate again, this is often a knowledge gap. We asked a lot, how do invertebrates respond to invasive species control or invasion? And that is a very legitimate question, as these form important part of the ecosystem, but it is a challenging question to answer. So this is Jacob Basso, who he's a really skilled uh, entomologist. He spent 2 years working full time on this. This is a photo here of some chironomids, but he could see through that scope. He counted every individual, and it was over 30,000 individual invertebrates. So again, I just wanted to add an element of realism of the amount of work that goes into this kind of monitoring. And we only identified them to the most feasible taxonomic level. So that is, we went to family, not genus or species. Because again, going down to species with chironomids is it's an incredible amount of work. And from our results, so this here is a panel of the density per meter squared of emerging invertebrates in invaded Phragmites habitat, remnant uninvaded habitat, and herbicide-treated habitat. The middle panel is taxonomic richness, so that's the number of different families per site. And the last panel is evenness, so that's how equal each family was represented. And what's interesting, what kind of surprised me, was that density was very similar between uninvaded sites and invaded Phragmites, but the density, the number of individuals in the herbicide-treated sites was very, very high. Similarly, taxonomic richness was somewhat similar between invaded and remnant sites, but lower in treated sites. And evenness was fairly low in treated sites. And this is because in herbicide treatment, we're seeing them really dominated by just a few families, in particular chironomids, so non-biting midges. In those first 2 years after treatment, you can see over the field seasons, this is going from May to July, just the number of chironomids was incredibly high in those herbicide-treated sites compared to the remnant marsh or Phragmites marsh. So this is really fascinating. Of the 30,000 or so total invertebrates that we captured, about 20,000 of those alone were chironomids. So lots and lots of chironomids emerging 1 and 2 years after herbicide treatment. And this actually makes perfect sense when we think about the ecology of these marshes. The treated sites had sparser emergent vegetation, and many invertebrates require vegetation to kind of crawl up out of and then emerge, like shuffle off that exuviate that you can see in these photos. Whereas chironomids are capable of doing that from the water surface. The treated sites also had very high water temperatures because they were just shallow open water wetlands. And there was lots of decomposing organic material from the Phragmites treatment. Chironomids, most of them are detritivores. So this really made a perfect conditions for really big booms of chironomids after herbicide treatment. And this is not necessarily a problem. Uh, we expect that the removal of the large amounts of plant biomass, that it does change the emerging invertebrate community. And chironomids are a very important food source, as you'll see in a few minutes. And um, we also looked at, which I didn't put in here, but we also looked at the aquatic, so the larval stages of these invertebrates, and they're fairly similar between um, the different types. So we expect as the vegetation grows back in, we won't see this big of a difference in the emerging invertebrate communities. But that extra food source was really a positive boon for those aerial insectivores, so swallows. Swallows were consistently higher in uninvaded marsh and in the herbicide-treated areas every time we sampled. So they were consistently foraging more often in the remnant marsh and in the herbicide-treated sites compared to Phragmites. And we expect this is because there's more prey in the herbicide-treated areas, and there's more larger prey items in the remnant marsh sites. But also, it can't be explained fully by prey. It's likely that foraging habitat that's optimal for swallows involves open water patches, involves that structural heterogeneity, the differences in vegetation. Um, so like size, structure, height, and the open water was truly a characteristic that was very important for a lot of these aerial insectivores. And we see that in the herbicide-treated areas and in the remnant marsh, we're creating more optimal foraging habitat, whereas the Phragmites, those dense monocultures, for whatever reason, they're just less preferential for foraging over. And I just wanted to point out another master's student who's currently doing work, Marissa Zago at the University of Waterloo. She's kind of picking up and looking more at the whole wetland bird community and how it's responding to the additional treatment that's going on in the National Wildlife Area and throughout Long Point to see how the bird community is responding. So she should have some results in a few years, and I believe she presented last week. So just a really brief research summary of everything we've done so far. And these are all published. Please feel free, I can share the PDFs with you if you can't access them. So the herbicide concentrations remained very low, below any level of concern in these sites, and they did not disperse very far from those points of application. So we can be fairly confident that this is not posing much of a threat to the ecosystems. And another important point, which Heather touched on, that I think is for any of you considering using a glyphosate-based herbicide, is that I think often the communication gets crossed here, which is glyphosate itself is the active ingredient. Glyphosate has been proven or demonstrated over and over again to be fairly non-toxic. However, as Heather pointed out, there's often surfactants added to these herbicides, and the surfactants allow it to stick better to leaves, similar. Some of these are proprietary, so we don't always know what goes in them, but this is the reason that if you buy Roundup over the counter, you cannot use it in an aquatic environment, and that is because the surfactant is what is toxic to the aquatic biota. So this is something that I found helpful when talking to people who are concerned about the toxicity of glyphosate, is that often the studies that we see, they don't account for the surfactant or what the surfactant is composed of. So when we see new research coming out saying, we determined Roundup was toxic to these aquatic biota, you want to always ask and you want to prime your potential audience or the people that you're trying to work with, you want to prime them to ask, did they look at their surfactant? What surfactant was being used here? Did they test that on its own for toxicity? And I just wanted to point that out because that's a lesson that we learned over and over again, and I found very valuable for communicating with the audience. Um, that glyphosate-based herbicide was really effective at reducing Phragmites, but in the first few years after treatment, we do see some secondary invasion from invasive species that were present in these marshes, and the vegetation community is somewhat sparse. It does take more than 2 years to really start to come back. And of course, the prevailing conditions of the site will affect that. If you have very high water levels, don't expect a meadow 1 year after you treat. And removing that established Phragmites, so managing and removing the biomass of very dense monocultures of Phrag, it will change the invertebrate community. You will see differences, and this is not shocking, not necessarily a negative, and you would expect that with longer term monitoring and the return of that vegetation community, that we will see the macroinvertebrate community begin to resemble native vegetation, what is in native vegetation in these marshes. And I just wanted to point out as well that restoration takes a long time and it can be imperfect. So these figures here are from Moreno Mateos from 2012, where they did a meta-analysis of global wetland restoration. And panel A on the one side is wildlife recovery, panel B is plant recovery, and that dashed line in the middle is reference. So these are wetlands that were never disturbed. And you can see that for wildlife and plants, it doesn't always get back to 100% perfect. Um, and in some of these instances, the monitoring went on for over a century after the restoration took place. And I think some people might see this and think this is a negative, but I think it is a hopeful thing to keep in mind because if we want to maintain these targets, if we want to preserve ecosystems, conservation and not touching things is not enough. We have to consider our relationship to that land and what we want to do to try to, or how we take care of it. So restoration is a really important piece in our toolkit. And even if it's not imperfect, it's better than only trying to parcel off a few pieces. Like a 70 or an 80% return is better than nothing. So I just wanted to highlight this here that for folks who might be discouraged or maybe read, oh, we treated Phragmites and then only frogbit came back, is that um, it takes a long time. These ecosystems, I mean, if Phragmites did not affect these ecosystems in a year, it's not going to go back in a year. Similarly, I guess I want to leave us all with the question of what is the purpose of our invasive species management? And I think it's important to consider this. Um, this was mentioned in the article that we don't want to ignore the impact of troublesome plants, but we want to determine they might be considered a plus to the landscape rather than an outright negative. I think a considerable amount of research has gone into Phragmites, so we know it's not a plus to the landscape. Uh,
5 minutes.
yeah, so we know it's not a plus to the landscape. However, some early um, work in the early stages of invasion did find when it was very low density patches, it wasn't necessarily incredibly detrimental to bird communities. So this means that even if you're working in places where eradication is maybe not possible because you would need to get every land user on board, and it does mean that we can consider continual management, long-term management of these invasive species at low population, very, very low population levels. That's not a failure. Um, Eradication doesn't need to be the only version of success. And I just think keeping in mind why we're doing this invasive species management, that column made me think about why are we doing this? And it's if we're seeing an improvement by controlling Phragmites, if we're seeing an improvement in native species habitat, then that is a bonus. That is a plus. That is excellent reason to continue doing this and to continue monitoring for multiple years. So I just wanted to thank you all. Here's my contact information. I've put a few more papers here that were recently published that I thought folks might be interested in. Where we've looked more into like niche differences and more into bird communities. But I just wanted to leave us on, I guess, the hopeful note of this works. It might take a bit longer, and I hope that everyone really does consider their relationship with this land and their role in conservation and stewardship and how that might affect how you approach invasive species on your own property. And I'd like to thank everyone who took part in this work and made it possible.